Welcome to all of you B.Sc. third year students in online lecture series of Fishery Science paper 13. We are studying the important aspects of our syllabus, marine water prawn culture or the culture of marine water prawn in seas and oceans. In last lecture we have seen collection of brooders, mating of brooders, then production of fish seed and hatcheries, how these are to be seen, how it is to be occur in this syllabus. See the figure. Figure shows these are the number of pinned prawns. These are the number of Pineus indicus. Shows the egg mass, which is to be in orange color. See, these are the egg mass which is to be in orange color. These egg mass, these are to be found in that is in thoracic region, or in these egg mass, these are to be found, these are to be occur, and this egg mass, these are to be found, these are to be occur. These eggs, these are to be found in these prawn, these are to be found in the Pineus indicus. In every prawn, show these are the some orange egg mass which is to be found, which is to be seen. Then for the culture of prawn, the next important article is the water quality for culture of prawns. To culture marine water prawns, to culture prawns in seas and oceans, to culture Pineus indicus and Pineus monodon in east coast and west coast, the prerequisite, the important requisite, that important requisite is the water quality for culture of prawns. Which type of water quality which is to be essential, which type of water quality which is to be required, which type of water quality which is to be ideal for the best production of the prawn in seas and ocean. For that purpose, for that condition, we have seen here, that is water quality for culture of marine water prawns in seas and oceans. Which type of water quality which is to be required, which is to be essential? The quality of water for prawn culture is probably the most important and crucial parameters. The quality of water for prawn culture is probably the most important and crucial parameter. Suppose any one parameter, any one factor of the water quality which is to be increase or which is to be decrease, very low or very high, then it cause the culture of prawn, it cause the crop of prawn. That's why these factors, these are to be most crucial, these are to be most important. The suitability of water in brood stock pools greatly influences the maturations of prawns. The suitability of water in the brood stock, those brood stock, these are to be transported, those brood stock, those are to be captured, those mature prawn, these are to be caught, captured for maturation. These brood stock pools greatly influence the maturations of the prawns. When these are to be stocked, where these are to be stocked, that water quality, these are to be important, that water quality, these are to be most influences on the maturation of the prawns. The quality must come very much nearer to that of the natural sea. Those areas in which prawns are cultured, those water in which prawns are to be cultured, that water, these are to be quality must come very much close, very much nearer to that of the natural sea water. Those quality of natural sea water found, those content, that is salinity, temperature, pH, turbidity, all those factors which is to be available, oxygen, carbon dioxide which is to be found in water, similar fashion, those parameter which is to be found in sea water, those culture water have a similar parameter or same parameter, these are to be found which is to be occur. The sea water must be filtered clear through the mesh cloth. The seawater must be filtered clear through the mesh cloth, sand filter or mechanical filter to remove the suspended impurities. Those impurities, those debris, those waste material, those unwanted material which is to be found, that unwanted material water must be filtered clear through mesh cloth, either by cloth. These are to be filtered with sand filter, or it is also filtered with mechanical filter to remove the suspended impurities, to remove the suspended materials or the waste debris and planktonic element which may be bloom and multiply later. Those water in which abundantly algal blooms, that may be red algae, that may be blue algae, that may be green algae. These algal blooms or the planktonic elements, these are to be more, these are maximum. Such algal blooms, these are to be keep in water. In later, in future, when water quality, these are to be ideal. These algal blooms, these are to be rapidly multiply, rapidly grow, and these are to be cover all water body, which is to be harmful for the culture of prawn. Hence, those impurities, those suspended materials or debris which is to be found, such as waste material, planktonic element, algal blooms, these multiply later. These algal blooms, waste suspended material, impurities, these are to be filtered by either through the mesh cloth filter, either through the sand filter, or either through the mechanical filter. These waste material, these are to be removed, these are to be discarded. Amounts of nitrogenous components such as nitrates, NH3 and nitrites, that is, these two important nitrogenous compounds, that is NH3 and NO3, these should not be exceeded and regulated carefully to avoid the larval mortality. That is, nitrates and nitrites, these are to be found, NH3 and NO3, these should be exceeded, these should be regulated carefully to avoid the larval mortality. When these NH3 and NO3, these are to be more, that is nitrates and nitrite, these are to be more maximum in water, larval mortality, these are to be occur. And once larval mortality, these are to be occur, prawn should be affected, prawn crop is to be affected. To avoid this, that is, these nitrates and nitrite, these are to be maintained, these are to be a proper manner. The seawater taken from different places will give different result. In culture practice, in culture purpose, those water which is to be you select, those water which is to be you choose from different places, different areas, different depths. These different places, different areas, these water give the different results. These water give the different result, means those areas which is to be your select, which is to be your choose, that areas, that water, these are to be a show the different result for the water quality. Following are the various factors which are good in proportions in the quality of water in the water for prawn culture. Following various characters, following various parameters, these parameters in an ideal way, in a proper manner, these parameters, these are to be found, these parameters, these are to be occur, then these parameters, these are to be shown in a proper way, in a proper manner. First parameter, that is, first parameter is this is the salinity. Salinity has a profound influence on the maturation and development of prawn larvae. Salinity has a profound influence on the maturation and development of prawn larvae. What is the salinity range? These are to be suitable. What is the salinity range? These are to be proper. The salinity range, these are to be 24 to 34 part per thousand. This symbol is the part per thousand symbol. These are to be studied, or this symbol, these are to be given. Part per thousand. And what is the salinity range? Salinity range is to be 24 to 34. 24 to 34 salinity range, these are to be seen, these are to be occur. And this salinity range, these are to be studied, these are to be discovered by which scientist? It is to be discovered by scientist Hudinga. These are to be studied that, and it is to be told that is those salinity range, these are to be occur for prawn culture. That salinity range is to be how much? 24 to 34 part per thousand, and this salinity range was found suitable for pinned larvae, which is to be discovered by scientist Hudinga. The salinity range varies for different species to species. For salinity range to the Peneus indicus is to be different. Salinity range is to be Peneus monodon is to be different. The salinity range varies for different species and should be determined by trials for the species selected for the culture purpose, means species to species. This salinity range, these are to be variable. Salinity range, these are to be different. For culture purpose, the first important factor for culture purpose, first important point, that is salinity range, and this salinity parameter, these are to be 24 to 34 part per thousand salinity range. These are to be maintained. These are to be proper way. These are to be chosen. Then next, temperature. Again we have seen that temperature, these are the seen. This temperature, these are to be seen. The temperature is an important factor. It is also called as triggering factor. Temperature is also called as limiting factor. This important factor in development, which is to be suitable for the species to species, which is to be verified by trial and error method. Which temperature these are to be suited, which temperature these are to be ideal for the culture of prawns. Generally, lower temperature may retard the development. Lower temperature, when temperature these are to be lower, when temperature these are to be less, then this temperature these are to be retard the development, or when temperature these are to be higher, it is to be accelerate the development of prawn. Means more the temperature, accelerate the development, less the temperature, it is to be retard the development. Means temperature range, it is to be neither more nor less, it is a optimum or a medium temperature range. These are to be select, these are to be choose, and this temperature range, these are to be selected. A temperature varying between 25 degrees centigrade to 29 degrees centigrade is considered to be ideal for the culture of Peneus indicus, for the culture of Peneus indicus and Peneus monodon. The ideal temperature range is to be select, and ideal temperature range is to be nothing but the 25 to 29 degrees centigrade temperature range. These are to be suitable to grow the prawns, to grow the larvae in a good condition, in a good manner. And this larval condition for the prawns, this temperature range, these are to be suitable, these are to be ideal. This is another important parameter, water parameter for the culture of the prawn. See, these are the various prawns which is to be seen in the instant figure. These prawn, these are to be cultured. These prawn, these are to be grown. These prawn, these are to be, when it is to be grow a larger size, marketable size, when water quality parameters, these are to be ideal. Water quality parameters in a good proportion rate, then these are to be grow, then these are to be increase in large size, then these are to be grow a ideal marketable size and get a maximum market fetching a good ideal price, which is to be best for useful for the prawn culture, is for the prawn farmers. Next important water parameter, that is the pH or hydrogen ion concentration. Again we all know that pH is nothing but the hydrogen ion concentration. The pH must be maintained at the optimum level. pH must be maintained at the optimum level. The optimum pH for the larval development has been found to range between 8 to 8.2. The optimum pH development, optimum pH for the larval development of the Peneus indicus prawn, the range is to be 8.0 to 8.2 and has been regulated carefully. The pH above 9 induce of phytoplankton. When pH these are to be increased above 9, which is to be induced, which is to be accelerate the development of phytoplankton or the blooms. When these blooms, these are to be increased, it is to be grow fast, then it is to be inhibited, it is to be affect the development of prawn. That's why above 9 pH is to be harmful, above 9 pH is to be not suited for the larval development of the Peneus indicus. Phytoplankton blooms which inhibited the development of prawn larvae. Water with 8.2 pH flowing continuously through the system. Water which is to be 8.2 pH, when it is to be continuously flowing through a system, it is to be influence the rapid maturation of prawns. And when these are to be constantly influenced, constantly flowing, then maturation these are to be occur. Maturation these are to be appear earlier as compared to the natural time. During recirculation and in bacterial oxidation of ammonia and nitrates, ammonia and nitrates, the pH was reduced, and so in closed system required quantity of water must be replaced daily. When ammonia and nitrate, these are to be increased. When ammonia and nitrate, these are to be more in water, at that time the bacterial oxidation, these are to be takes place, and during the recirculation bacterial oxidation, ammonia and nitrate, these are to be increased due to the less pH, minimum pH. At that time, such time the water, these are to be recirculated, water, these are to be changed daily, water, these are to be changed daily, old water, these are to be removed, leave out, and new water, these are to be added. In that water a proper range of pH, these are to be maintained, and that proper range of pH is 8.0 to 8.2 pH range, these are to be maintained. It is also another important physical or the biological especially hydrobiological characters which is to be maintained, parameter which is to be maintained for the culture of prawn culture, culture of Penis indicus. Another interesting character which is to be we have seen, that is the light intensity. Another interesting character is to be seen, that important character is the light intensity. See light intensity, why this light intensity, these are to be interesting, because for the experimental studies it is to be studied by different investigators, different scientists, and shows that experimental studies have revealed that the larval development proceed in calm and darkness. Larvae which is to be present, larvae of Penis indicus, Penis moundon, these are to be grow, these are to be developed well when water calm and darkness. In natural way, prawns breed at bottom of the sea, where light is to be very low. Naturally prawns, these are to be breed, naturally prawn, these are to be spawn in the bottom of ocean, bottom of the water bodies of the seas, where light is to be low. Strong light in maturation tanks may be source of stress. When heavy or the dark strong light, these are to be appear in water, at that time those larvae, those prawn, these are to be found, these larvae, these prawn, they can stress, they can frighten, and they can affect the development, affect the growth. When inner surface of tank is painted white and prawns dash against the walls get injured. When we have painted inner side, when we have painted the inner tank of the white paint, at that time these prawns, these are to be dash against the walls, against the tank, and they can injured. Their legs can be broken down, ciliate legs, thoracic leg, these are to be cut down, and they can remain injured. To avoid this injury, water becomes calm, quiet, and it is to be dark condition. It is because of this that tanks are covered with shades and their inner walls are made to painted a black painting or the black color. This black color, these are to be used to painting the inner side. When these are to be painted black, prawns, these are to be do not, that is specially these are to dash the dike or the bottom, and they do not injured, and they safe. That's why inner side of the tank, these are to be painted with black paint. It is to be suitable, it is to be ideal for the culture of prawn culture. Another important character, dissolved oxygen. For the survival, for the growth, the important parameter, that is the dissolved oxygen for the development, for the growing. Dissolved oxygen, optimum oxygen, dissolved oxygen of water for larval development in different species ranges 5.0 to 9.2 ppm. The dissolved oxygen level is to be 5 ppm to 9.2 ppm is to be ideal, suited for the growth of prawn. The carbon dioxide should be nil or minimum. At the same time, when at the time of dissolved oxygen, these are to be maintained. At the time of dissolved oxygen, these are to be proper range in 5 to 9.2 ppm. At the same time, the carbon dioxide level is to be nil or it is to be minimum. And when carbon dioxide level, these are to be nil or minimum, then these prawns, these Penis indicus, these are to be grow better or accelerate a ideal way, ideal manner. This is another, that is water parameter. Next water parameter, that is pressure. Prawns are demersal in habitat, and naturally it is presumed that pressure might influence the maturation and spawning. Prawns are demersal, it is to be found in deep water. When it is to be found in deep water, demersal in water, inhabit, and naturally it is presumed that pressure might influence the maturation and spawning. Sometimes pressure these are to be occur, sometimes pressure these are to be affected on the, these are to be presumed influence on the maturation and spawning. On the spawning and maturation, pressure these are to be affected. That's why immature prawns spawn in bottom of tanks without applying pressure. Hence, for the prawn culture, the pressure or the atmospheric pressure these are to be normal. It is to be on sea level or MSL level. It is to be 1 atmosphere, or on a general 1 atmosphere pressure these are to be affected. These are to be seen. Then and then these are to be grow. These are to be well better. This is another important pressure. These are to be more the pressure. These are to be affected, as well as minimum pressure. These are also affected. That's why 1 atmosphere or the sea level atmospheric pressure these are to be suitable for the culture of prawn. Then another important parameter, other chemicals. Not only the dissolved oxygen, pressure, light intensity, pH, salinity, but other chemical these are also important. Other chemical these are to be most useful. Which are these other chemical? Accumulation of ammonia and nitrites. Ammonia and nitrites in culture tanks is toxic to the larvae. Accumulation of ammonia and nitrite, these are to be toxic, these are to be harmful, and these toxic, these are to be harmful to the toxic to the larvae. Older larvae are more tolerant to the metabolites. Those larvae these are to be old in size. Those larvae these are to be old in age. These larvae these are to be more tolerant to the metabolites. The broodstock prawns require lower concentration of ammonia. Broodstock prawns these are to be require lower concentration of ammonia and nitrites for gonadal maturation. For the gonadal maturation in broodstock, lower concentration of ammonia and nitrite it is to be ideal. When these ammonia and nitrite these are to be in lower in maturation ponds, in maturation tank, at that time these prawns these are to be mature and these are to be spawn. Means not only the other factor, but these nitrites, ammonia, these nitrites, ammonia, these are also affect on the metabolites. These are to be affect on the spawning. These are also affect on the development. All these are to be cause and maturation. Hence, these other chemical, other factor, these are also important. These are suited for the culture of prawn. Means when we have select, when we have choose a water, this water or that water must be maintained pH, must be maintained salinity, must be maintained the intensity of light, must maintain the dissolved oxygen level, must maintain the pressure and other chemicals. When all these are to be in ideal way, in a ideal manner, then and then our prawn culturing these are to be suited, these are to be done very well. Otherwise, any one factor of these parameter, water quality parameter, these are to be imbalanced. Any one factor, these are to be not in a proper proportion, not a proper level, then the crop of prawn these are to be lost, and prawn culture these are to be imbalanced, prawn culture these are to be disturbed, and when these are to be disturbed, the expected prawn production these are to not occur, and when expected prawn production these are to not occur, then it is to be harmful, it is to be important, it is to be a risk factor for the prawn culture. That's why when we have culture prawn in sea water, in ocean water, marine water, first we have maintain in mind, first we have keep in mind, that is the water quality parameter these are to be in proper range, in proper way, then and then the prawn culture these are to be successful, these are to be a good ideal. With this we have seen the water quality parameter for the culture of marine water prawns, culture of sea water prawns. With this again we have completed the other aspects of this culture of marine water species or the culture of marine water prawns. Thank you.